Hello students, today I am going to teach you a science subject. In that I am going to teach you a topic cell and cell organelles. A cell is the structural, functional and biological unit of an organism. Cells are the basic building blocks of our muscles, bone and other organs. In 1665, Robert Hooke discovered the first cell. He was an English scientist. He used his microscope to look at cork cells. And the next topic is components of cell. Cell wall is also called as a plasma membrane or an cell membrane. It can be sealed by both the types. Cell wall is the outer covering of a cell present adjacent to the cell membrane, which is also called the plasma membrane. The three major layers are primary cell wall, the middle lamella, and the secondary cell wall. Mitochondria was first discovered by Colliker in 1850. Each cell requires energy, and this energy is produced by mitochondria. The outer membrane is porous and the inner membrane is deeply folded. These folds are called cristae. The inner cavity is filled with proteinaceous gel-like matrix containing ribosomes, phosphate granules and DNA. Therefore, it can produce its own proteins. With the help of enzymes, mitochondria oxidize carbohydrates and fats in the cell. The energy released in this process is stored in the mitochondria in the form of ATPE, that is adenosine triphosphate. Plant cells have less mitochondria than animal cells. Mitochondria. The components of mitochondria are divided into four parts, that is inner membrane, outer membrane, cristae and matrix. These are the four components of mitochondria. We can see nucleus under an electron microscope. We can see nucleus covered by a double-layered membrane with small pores. These pores allow the passage of material in and out of the nucleus. Nucleus has one round nucleus and a network of chromatin fibers. Chromatin fibers are thin thread-like structures which condense to form short, thick chromosomes at the time of cell division. Functional segments on chromosomes are called genes. The function of nucleus are it controls all metabolic activities of the cell and also the cell division. It is involved in the transmission of hereditary characters from passenger to offspring. Due to loss of nucleus, large quantity of hemoglobin can be accumulated in the RBC and thereby large amount of oxygen can be transported. Do you know? Due to loss of nucleus of sieve tubes of the plant phylum, they become hollow and thereby transportation of food become easy. Lysosomes are simple single membrane bound sacs filled with digestive enzymes. The first function of lysosomes are immune system. It destroys viruses and bacteria that attack the cell. Second function is demyelination squat. It destroys worn out cellular organelles and organic debris, that is autolysis. The third function of lysosome is suicide back. When a cell becomes old or is damaged, lysosome burst and enzymes digest their own cells. The fourth and the last function of lysosomes are during starvation, lysosomes digested stored proteins and fats. The organelle which conduct various substances inside the cell is called endoplasmic reticulum. ER has a net-like structure consisting of interconnected miniature tubes and sheets filled with fluid. ER is connected to nucleus from inner side to plasma membrane from outer side. Rough ER has ribosome granules on its outer surface. The functions of endoplasmic reticulum are: it is a framework that supports cells, conduction of proteins, and toxins that have entered the body through food, air, and water are made water soluble by air and then flushed out of the body. Golgi complex. It is made up of 5 to 8 hollow and flat sacs called parallel to each other. These sacs are called cristae and are filled with different enzymes. The proteins coming from ER are enclosed in vessels which come towards Golgi complex via cytoplasm. This fuse with the formation phase of the Golgi membranes are empty. Their contents in the cristae. As they pass through the cristae, they are chemically modified with the help of enzymes. They are again packed in the vesicles. These vesicles exit from the maturation phase. Thus, cristae work like a packing department that pack and distribute substance. Vacuoles are the storage sac for solid or liquid contents. They don't have any typical shape or size. The structure of vacuole changes according to the need of the cell. Vacuole is bound by a single membrane. The function of vacuoles are to maintain the osmotic pressure of the cell, to store metabolic byproducts and end products, that is glycogen, protein, water, etc. In animal cell, they store waste product and food, while in amoeba it store food before digestion. In plant cells, vacuoles are full of cell shape and provide turgidity, rigidity to them. Chloroplast. Chloroplasts are important for the photosynthesis process that take place in the leaves. Chlorophyll in chloroplast traps solar energy and convert into chemical energy. Stroma in the chloroplast contain enzymes, DNA, ribosomes, and carbohydrates that are necessary for photosynthesis. The functions of chloroplast are to convert solar energy to chemical energy. Chloroplasts give different colors to flower and fruit. Nucleoplasts are involved in the synthesis and storage of food like starch, oils, and proteins. Thank you everyone for watching my video. I hope you have understood the lesson. Thank you everyone.